this video we will discuss about this FOXP3 regulator using perturb-icCITE-seq analysis. So we have discussed in our previous video that how they made this FOXP3, how they make this CRISPR whole genome CRISPR knockout libraries and use it to find out the genes that cause these FOXP3 upregulations and downregulations. Here they did the FOXP3 they check out or find out the FOXP3 regulator using perturb-icCITE-seq. So how they did their studies is they took the primary T cells, that is CD4 plus T cells, and then transduced them with these 905 different guide RNAs, and which targets the 295 different gene knockout. And thereafter they did the electroporation of this CRISPR libraries gRNA infections, and then the edited cells will be edited T cells will be activated. That is, T cells IT-reg inductions will occur, and then check for the surface protein staining around 277 proteins, cell surface proteins, and then check for the perturb-icCITE-seq intercellular results. And what they are able to find out is that the results that they find out is that the FOXP3 expressions goes down, on in some cases goes up. For example, when the RBJ knockout is there and ZP3B knockout is there, it will goes up, whereas when the FOXP3 knockout is there, it will goes down, which is evident. So this, and when they check it for CD25 expressions, they can see that with the knockout of STAT5B, the CD25 expression goes down, but with the knockout of STK11, CD25 expression goes up. Similarly, when they check it for IKZF3 protein expressions, it goes up. When this ZBTB7B knockout is gene is knockout, and also when the P10 gene is knockout. So like this, they did the cell surface or intercellular studies of this different guide RNAs knockout, and thereafter this is the overall expressions to check the FOXP3 expressions, as most of these genes are will cause down regulations in the FOXP3. So they can see that FOXP3s are being down regulated in some cases, and in some cases it has been upregulated. So this knockout libraries not only, let me correct myself, this knockout libraries not only target the one kind of regulators that can either upregulate or downregulate. It is the mix of guide RNAs, and they want to find out which genes cause upregulations and downregulations of this FOXP3 protein expressions. They can see that some of the proteins cause upregulations and some of the proteins cause downregulations. And now here they plot this heat map where they plot the FOXP3 expressions with different perturbations. For example, we can see that the perturbations can be seen. Perturbations modules can be seen in this vertical directions, and the gene program can be seen in the horizontal directions. And we can see how the FOXP3 expression changes in this case. And we can find out that the analysis of this has been shown in this panel E, where we can find out the different modules are been regulated by different sets of genes, that is mentioned in this panel E, which they have mentioned which modules are being regulated by which kind of genes. And interestingly thing that they are able to find out is that module G is regulated by only this one kind of gene, that is ZBTB7B, and they also able to find out that this RPPJ gene will cause upregulations of the FOXP3 protein. So this is what they are mainly able to find out that. And when they plot this map that is on the right hand side of the panel E. In this they initially used to 27 knockouts, and thereafter it combined them into the 10 knockouts, and these modules that will has an impact on this FOXP3 protein expressions. So out of this they are able to see that the RBJ has a role towards upregulations of the FOXP3. So they plotted a map where they are able to see that they plotted a map between the control and the RBJ knockout, and they are able to see that RBJ's levels are low and the FOXP3 expressions are high in that case. And on the left hand side is just the continuation of this panel E, where they defined different modules and what are the genes that are considered in that modules. So I hope you understood. And in this case, in the activating and inhibiting are represented by red and green. So the FOXP3 expressions are being upregulated by the lines that are connected through this blue, and the inhibiting is regulated by the lines that are connected through blue, and the activating FOXP3 positive regulators, we can see that, are being connected through the red lines. So this is how we can read this map, and this is just the module descriptions that are being used in the that information is being gathered from the panel D, and which are being used to plot this left hand side or right hand side panel of this panel E. So I hope you understood how these FOXP3 regulators are identified using perturb-icCITE-seq. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.